Well, greetings everyone. Tips again. Time to have a discussion about trees, insects, pests, and soils, and see what we can come up with that satisfies our appetite for further information. And last time round, a participant sent in some questions and said, "Do we have we discussed it?" And I said, "Don't think so." Peter's confirmed that we haven't. So I would like to share that with the group and answer some of those questions. And there's some really good stuff in it, in the questions that he's asked. So let's just see if I can share that now. Share screen. I should be able to share my whole screen. And you should be able to see a Microsoft Word, an image. Can you guys see an image in front of you?
Yeah, it's working.
Okay. So these, the person that sent these along was whom? Let's see if I can find out that for you. Nissi Carlson. Okay. So first question you've got for me, and you need to have some dialogue with me on this one.
Yeah.
There looks like there's a crack in the bark, doesn't there?
That's what it looked like from the ground, but I wasn't sure.
Yeah. Now, look, I think it has been loaded, and that looks like it's a part of what we'd call an oblique fracture. So it's gone up at an angle.
Yep.
And that's not necessarily a problem. No, it's horrible when I say that to people. But trees have got this ability to stitch themselves back together, much the same way as bone does, except trees can only do it from the outside. Bone does it all the way across the surface. And the amazing thing is that doing it from the outside still gives incredible amount of strength. And I was just reading a paper today that looked at the effects of carbon on global warming. Carbon dioxide on tree strength, and said, trees with increased carbon dioxide grow faster. The wood's not as dense, and that's because it's growing faster. It's putting on a lot more, in particular, early and mid growth wood rather than late wood. But the one thing that it didn't consider was this modulus of rupture or failure, and that is the further you move things out, the stronger they become. So a hollow tube that is a long way out and 3 mils thick is a lot stronger than a 6 millimeter solid rod that you can just bend. So the further you move that 3 mils out, the stronger it becomes. And so the tree is going to start stitching on the outside if the crack has gone through into the wood. It may just be in the bark. If you were concerned, if it was in a higher target area, one of the things you could look at doing is doing some reduction pruning.
Sorry, that limb, or that's actually half the tree sort of thing, is leaning over a living room in a house. And I went up. We went up in the cherry picker and had a look, and it turned out it was just the bark. But I definitely was concerned when I saw that from the ground.
Yeah, and so this is this process of learning and experience. So the bark fails on occasions before the whole stem. There's a fair amount of flex. Torsional cracks in particular, you'll see it, and it looks spectacular. You'll see these spiraling cracks in the bark as it twists. And you can do this if you've got a banana, and it's one of Frank Rinn's favorite examples is if you get a banana, you know how when you bend the banana, you crack it and you open it. Well, that's a hard way for a kid to do it. If you've got a kid or a grandkid, get them to take the banana at either end and twist it. And you'll see that torsional failure is really easy to occur. And wood's not as strong torsionally as it is in tension.
Yeah.
Of course, this wasn't in tension. This is in flex again. We talked about that once before in one of the tips. And so it does fail when it's loaded beyond the extreme. But yes, look, even overhanging a living room, the first thing I would say is, how was the house constructed? How big's the part? Realistically, do I expect this to go through a living room and squash a person? Or is this just a household insurance claim? And if it's a household insurance claim, it might be $10,000 or $20,000 worth. Boy, it's one of the cheapest renovations you'll ever get done in the house. So you've got to weigh that up. Is it necessarily something you need to panic about? and it might not be. Let's just say we think this has got a significantly increased likelihood of failure. We might put that at somewhere in the 1 in 100 to 1 in 1,000 likelihood of failure. You know, that's a fairly high likelihood of a part failing. And we say, well, it's going to do $50,000 worth of damage. Well, that's 100 into 50,000 is $500 a year. Well, no, is $500 a big amount? Not really. Maybe, you know, we can live with that and see what it does. And I've certainly confessed to leaving trees like that in schools in low use areas, simply because, you know, the odds are statistically well and truly in the favor of the tree not doing anything. And I'll have a look at that. If I can, I'll share an experience of that in the last month that was really quite surprising to me, where I decided to look at the statistics of the use of a school playground and was floored with how infrequent the use was. So that's good one. And this is the tree itself.
Yeah, exactly.
Yeah.
So that crack is above that main union there.
Yeah. So this is not exactly what I'd call a big part.
No.
Metal roof, wooden or metal framed building. It probably would have done maybe $500 or $1,000 worth of damage if it hit the roof. And so I would have looked at it and said, look, you know, run the punt on it if you want. It's up to you. And, you know, I'm not the consent giver. So, you know, if necessary, you're happy to go back to the council and ask the council to make a decision and let the householder make the choice. But most of the time I spend encouraging householders to take some element of risk. We drive a car, you know, that comes with an element of risk. We, you know, I fly to America, or I drive to Queensland, and the one that poses the greatest risk by a factor of 4 or 5 is the trip to Queensland, not the flying to the US. So, yeah, it's an interesting one. So yes, good one. Now, this is a fun one.
I thought you'd like that.
And this is a nice little round ball. And this is an epicomic growth that's coming out of that round ball. And this is really the equivalent of a burl. And there's a great little name for these called a Sphaeroblast. And now I know I've talked about this once before and tried to find the information at short notice, but the fact that you'd sent me the picture makes it easy because I'm prepared, and so I can give you some information on it. And I think that's great. And if we look at how meristematic tissue develops in these, there are these things called bud traces, and I'll see if I can open one for you. Bud trace. I may get it through the search for bud trace. No. Let's try burl. Oh yeah, here we are. So this is an Araucaria Sphaeroblast. And this here is the meristematic trace, or the tissue that was going in all the way to the middle. So these traces start oftentimes at primary growth, and they'll run in Araucaria, for example, run for 50 or 60 years in age, very long-lived. If you look at something like Cupressus or Cupressus apyrus, or wherever we want to fit it at the moment, those latent green hedges, they might last for 5 or 6 years and then they die. They're eliminated in the growth process. So these traces enable epicomic buds to develop. Now, they're not a bud as such, because a bud needs a leaf primordia. It needs a little printing machine at the end. But this is specialized meristematic tissue that is just so predisposed to do things that it's used for propagation. Redwood sphaeroblasts, quite commonly used to grow redwoods, Sequoia sempervirens, apples, similar, pears, olives. So they're highly meristematic zones. And when they come out, these little round balls, lift up the bark and you get the tissue changing. And if you look here, you can see there was a ball there and a ball here and a ball here and a ball here. And here's that ball with that little trace. So this is an Araucaria burl. And this was a sample that student Jeff Ashton pulled apart and developed. It was a really brilliant teaching sample. We've long since lost parts, but again, you can see that's a part of the ball, and as it pushes the bark, that tissue is no longer in ring tension, so it divides rapidly. And I know we've talked about this previously where I've said, and I think you can see my hands, if we've got our rays, they're held in place. These traces are held in place by the adjacent tissue. If we put pressure on them, they spread and they open up. So we've got the diamonds that Manic talks about. If we put pressure on that, it opens up, they herniate to the other side, and then we get this highly meristematic tissue wanting to divide rapidly. And so it starts to divide rapidly. And when it starts to divide rapidly, we get burls, and we get one meet up with another and so on. And that's that lovely effect that we get. And there are other causes of sphaeroblasts or these aberrant growths where it's done by a fungi or done by insects. We have a particular name for it. It's called a gall rather than a burl because it's initiated externally. If you're English, they're called burrs, just to mess with everyone. Nobody's got common terminology. And there's not a hell of a lot of information on them. You can go do Google Scholar and you'll be in and out of it in 200 articles of that, and most of them don't have a lot. Same with the books. And I started a search for you, Niels, a few hours ago to find out how many times Sphaeroblast occurs in my digital library. I've got about 5,500 books in the digital library. And this is it here. It's giving the books and then a hypertext link to them. And you'll see that some of them aren't different and some of them are things that don't matter. So this is a rhododendron species, Sphaeroblastum. So it just, Sphaeroblastum literally means circular bud. Okay, so a sphaeroblast is simply saying it's a round bud. And so there's not a lot so far. I searched for 6 hours and 27 minutes. I've searched the entire library. Half of these are duplicates, so that they're there more than once. Here we are on olives. So, and this is a neat piece of software called PDF Exchange Viewer. You can download it for free. It works on the Word or Microsoft platform, the PC platform. I don't think it works on Macs. Nothing works on Macs except Mac. Peter's having a laugh with me at it. But the nice thing about it is once it's finished searching, it gives you these hypertext links. And just by going like that and clicking on it, it opens the page up in the PDF document, highlights the word, and you can find it straight away without having to do anything more. I'll just enlarge that to 150% so you can see it. And here in olives, juvenile shoots are obtained from sphaeroblasts. Okay, well, that's great. That didn't tell us that. It just says it's for propagation. If you start looking through, you'll see there's a lot of stuff there on propagation. But here's the next text, and there we are. It's from a PhD thesis. And so there's not a lot there that gives us anything about these wonderful things called sphaeroblasts. Same when we go to Wikipedia, and I did that earlier on. Hopefully I can do it again. Control F.
Sorry, Mark. Are they a result of load on the tree most of the time?
Yes, that's my my understanding of the situation. It's a result of them herniating through the constraint of ring tension. So,
yeah, it's quite common on sugar gums, and it makes me think: is this particular tree I'm looking at experiencing a lot of load because it's showing these all over the trunk?
And you'll often see them in a line.
Yeah.
Particularly on things like ficus, for example, very commonly seen in lines of platanus again, same thing, in these sort of rows of things. and often they don't do. They're of no consequence.
No.
Other times it's the genetics of the plants being played with. So bacteria and viruses can have an impact. But, we often see them. If you knock the bark off a eucalypt, you'll see what appears to be thorns. And the thorns are where it's been constricted by the adjacent tissue and it hasn't been able to herniate out.
Yeah.
But you'll frequently see them herniating out. So now let me see if I can spell it correctly, because it's a horrible spelling. Sphaeroblast, I think it is. And that's where I got it wrong last time. No, SPHAE. Yeah, there we are. So, that's what we're essentially looking at, these nice rounded structures. And whilst that's a nice thing, it is missing so many references and support that it's just of little value to us. And that's generally what you'll find. Again, if we go to Wikipedia, to Scholar. And so if you want to do a PhD, it'd be a really easy area to get yourself a PhD in. Again, completely irrelevant. No one wants to study them. They've got some advantages, but there we are. 260 results total. And when you consider some of them relate to diseases and others relate to propagation, that's not a lot in all the scientific literature. So,
hey, Mark.
Yes.
Sorry to interrupt you there, mate. My name's Joe, by the way. Sorry, I haven't got my camera on.
I see you. I see your name.
But look, cool. Look, I was just wondering, and I joined a little late, so apologies, I might have missed it. But you mentioned before about how similar thing which is created by insects is a gall, which I'm fairly familiar with, but I might have missed it. The difference between a gall and a sphaeroblast is effectively if it's created by an insect, is there a way of telling whether it's a sphaeroblast or insect created gall other than cut it into it? I might have missed it or misunderstood.
No, that's a really great question. Almost all of the young sphaeroblasts are pretty easy to pick that they're sphaeroblast because they're almost circular and you can pop them off the tree. Often called tree eggs as a nickname. They've got this very little, that little meristematic trace you saw. I don't know whether you saw that in the picture earlier.
Yeah.
And so they pop off the tree. They're not held on very securely. So these you can just come along with your fingers and just pop them off, or the toe of your boot, just give them a quick clip and they'll fall off.
Yeah, because I've seen a lot of the galls which I generally tend to more associated with sort of on smaller branches, etc. But yeah, it's funny. Now you sort of brought it to my attention. I've noticed these shapes a lot on araucarias, which I think you had an example of before, and I've always wondered, okay, is that a gall or what? So yeah, it's interesting. I just wondered whether if they're off the trunk, is that more typically, I'm guessing, non-insect related as well?
That's right. And if this was an insect, there'd be several things that we'd be looking for a gall.
Mm-hmm.
If it was a hemiptera gall, because hemiptera don't ever have chewing mouth parts, there'll be a small little opening. And so if I can go back to some images, galls always are going to have some form of an opening of the hemiptera. Let's have a look at those.
Yeah, okay.
So that leaves us with only several others. So this is magnified a fair bit, but there is a opening to the gall. This one's causing dimpling on the trunk. Again, here you can see we've got these internal chambers in the wood.
Yeah, it seems quite obvious now. You're sort of showing the two, but
okay. But here's where it starts to get difficult. This is a wasp gall. Now it's on a leaf, but because the wasp has a chewing mouth part, it's Hymenoptera, so its adult stage can chew. It's able to chew a hole out, so you don't see it. But this none of these insects will generally live more than a couple of years. The exception are the hemiptera. Some of the hemipteras will do 3 and 4 years, but most of these guys, 6 or 12 months and they're out and moving on. And so you'll see a chewed out hole rather than a round hole. And so it's the hole in the gall that'll give it away. Here's one on a branch, and you can see the same thing. The branch has got all these little wartiness that's created, and yet that's sort of a fairly even, not chewed hole. So we know that's a Hymenoptera gall of some description. And you'll find this amazing. Half these things haven't even been named yet. There's an entomologist, a brilliant Australian entomologist, Penny Gullen, who studies these, along with, and I can't think of Dr. Lynn's surname, out of Queensland. They just love these guys, these unusual group of hemiptera and scale insects. And trust me, it's easy enough to find a new one that they've not seen. Because, again, who's interested in something that does that to a bark of a small branch? Almost nobody. And so there's lots of stuff we still don't know. And the response from the arborist when the arborist saw this is, let's just cut the tree down. My response was, boy, we should try and get this identified. The tree went before it got identified. So that's that. Does that help you figure out that they're slightly different? And then there's things like this. This is a gall again. This is on Casuarina. Casuarinas have some really fascinating associations. But this is just, again, altering the tissue. So these are, again, we saw, I think last time, these are the articles of the scales, and this particular insect has made those leaves fill right up and become super plump using the chemistry of the plant, rediverts the chemistry. And then it's subsequently been attacked by another insect, probably Hymenoptera. And then we've also got mites on it as well. So, there's complex things going on in plants. Just amazing. So, yeah, I don't think there's anything perfectly easy. Burls tend to be more irregular in their shape. So, here's there's burl common on, and all this is a high polymeristematic tissue. If that was down low at the ground, like we see in the one behind here, that's a lignotuber on a Lophostomen convertus, and they're really common on Lophostomen convertus. Some Lophostomen don't get it, and I suspect they're from areas that have been moister. I think what happens is when you get fire go through, you end up with plants that tend to be more capable of regenerating from fire. And so lignotubers are very important for those.
And it's crazy with the lofos as well, like the difference. You can have a whole avenue lining a street, and just from one tree to the next, the difference in the amount of that sort of swelling and lignotuber they have at the base. I've been doing a lot of audits for Strathfield Council, and they've just got avenue after avenue and of lofos. But it's
knowing what we know now, if we were really smart and cluey, we'd start selecting for non-lignotuber strains for street tree plantings, because they don't get anywhere near as big a trunk diameter. We don't select on that basis, and it's a shame. But we could. I guess I'm sarcastic about how nurserymen do things. I think the way nurserymen propagate are looking for things that have failed on the ground and say, yes, we'll propagate from that because it's easy. I know that's not true. But, it's like Melaleuca quinquenervia is a beautiful street tree, but the trunks get too big. I think we can agree with that for most plantings. There's an aureate form of it that occurs naturally. You'll see it planted or growing naturally in street tree plantings in Sydney, that grows at probably 60 to 70% of the speed of the green form. It seems to me that we probably should look at using some aureate forms just to get slower growth, or better still, look at something like Melaleuca decora or linderifolia or something like that. that again is smaller growing. But you can see here, you know, this has got massive amounts of burls. There's probably some other contributing factor apart from just load in that. And one of those things is going to be some genetic propensity, just like there's genetic propensity to all sorts of things out there. Interestingly enough, someone wrote in an article, I was reading it only the other day, that the largest burl in the record was just over 2 and a bit meters long. And I can assure you there are burls that are a lot bigger than that in size out there. I know I've seen one that's probably closer to 4 meters because I thought about it as a dining room table, but national parks would have probably been disappointed with me. And that's a problem. People steal them. People like them from their turning point of view, and they do look great. So, you know, there's an araucaria with all sorts of wonderful lumpiness going on. That's mudgy. That's a Lagostromia. That's Crimbia maculata. And here, the burl has created some problems. As the tissues started to include on itself, it's actually started to kill areas off. So this one is one that's been quite, in the end, destructive for the host tree, which is a shame. So that's your Sphaeroblast. And as I said, unfortunately, I can't give you much information on it. By the way, that's the same tree a little earlier. You can see how it has started to degrade from that earlier shot. And there's what we were really looking at. This is in a Kauri in New Zealand from the Kauri Museum. You can see the same burls developing quite naturally. A wonderful process. Okay, so back to your pictures. Niels, does that answer the question you've got on?
Yeah, it did, and then some. That was really interesting.
Yeah, look at it. It's a great little area. I don't know. I closed it down, did I? Must have closed it down. Oh, no. Okay. It opened it. Don't ask me why I did that. It did it as a Word document and then opened it elsewhere. So, yes, they're great little things. Okay. Next question. What's this fungus that we see here? And the color of it alone suggests to me that we're probably looking at Genoplis. We're getting no lesion. We're getting no inverted wound that says tissue's dying off. It doesn't look like it's pathogenic. Normally, you start to see some dying off if this was armillaria. And that's the close-up of it. And it's not easy enough there for me to say, but it's starting to look like it's got a bit of brownie coloring in it. And that browning color is because the spore is rusty, orangey brown on Genoplis. So my gut level feeling, based on the images alone, is Genoplis. The way I would have tested it, grab a pea-sized piece, chew. Chew for about a minute. If it gets hot and spicy like chili, malaria. Okay? If it doesn't, it's not. The next easiest way is to take one of those caps, put it down on some half black, half white, or some form of paper that'll allow you to see various colors. Put a cup or a mug over it for about 12 hours, so leave it overnight, and have a look at the spore print. Armillaria is white to slightly creamy. Genoplis is brown, and that at just on a visual looks like Genoplis.
Do they appear at different times of the year as well, or are they always just the same time?
No, Genoplis has got a lot more extended period for producing sporocarps, so it's a lot harder.
That's a really shit picture, but there's
but it's a picture that's still got ability to tell us some things. We've got an insect, it's a beetle, got these hardened wings called elytra, long body shape. I'm guessing it had long antenna at one stage. And
these ants have made a complete mess of this girl's whole backyard. If you scroll down a bit, you can see the other pictures. There's just the whole backyard is like that.
Yeah, and look, ants are wonderful things, and I don't want to be horrible to them, But the white-footed house ant is a really nasty ant that likes, it's an invasive species introduced, and we get them in the house. They'll come in during summertime and they'll have big heads and little heads. So you get the big-headed worker and they're just ugly. And then you get smaller ones. And again, I'm pretty sure I can do this. They can get huge colonies. So I'm on 5 acres. We've got about an acre under concrete. I apologize for the fact that I do have that. And we had a massive infestation a few years ago. So we just used some standard ant baits. And here are the carcasses they pushed out. And we treated, and we're getting impact 100 meters away from the treatment. That is, the colony was a single colony that extended over 100 meters. This gives you an idea of carcasses just being pushed up. They don't want dead carcasses in their colonies, so they push them to the surface. And you can see just en masse. And this extended, as I said, for more than a kilometer, more than 100 meters over the property. And so ants really do have an important role in plant life. We don't want to get rid of them necessarily. So the first thing is identifying them, and that's hard. There's very few that are truly remarkable. This is one of them. This is the muscleman ant. If someone can find me a colony with some reproductive queens, I'd be happy to look at negotiating a purchase. They are delightful, very friendly, in spite of their huge mandibles and massive size, very docile ant. And they're carpenter ants. They're an ant that goes in and chews out wood from the inside of the tree. They also farm, interestingly, scale insects on the inside of the tree, which is even more amazing because the tissue they've got to connect to, the scale's got to connect to, has to be living tissue. So they've got to cut away the wood from the inside until they reach live tissue. So these are truly spectacular, and they're one of our more primitive ants. So yeah, definitely great creature. Peter, you'll remember these. We exposed a colony in class.
Yeah, I do remember them.
Yeah, great guys. So if I'd known what I know now, I would have done a lot better job of keeping them and maintaining them. But at the time, my knowledge of rearing ants wasn't so good. And so you get the idea of how sick people are with their research. These are ants that have been coloured up so that they can see what they do. So someone's done some PhD research. And there's an even more fascinating ant that has a see-through body that they can colour up and give colouring to. So, and if you haven't seen it, there's also a process, and I noticed yours there, Niels, was on sandy soils. Are you near a coast somewhere?
I'm in the Grampians. I guess it's fairly sandy there too.
Yeah,
actually. Okay. It's all sandstone.
They pour molten aluminium into the ant's nest to make sculptures.
Oh, I've seen that actually.
They're really quite brilliant. So these sugar ants have been fed drops of coloured water, and then it ends up colouring their abdomens, and they can change the colour based on the diet that they're having, which is pretty amazing that somebody sits there and thinks about this sort of stuff as research. So yeah, again, I remember many years ago being over in Bartlett Labs, having a discussion with one of the entomologists, and he had named several insects, and I was just amazed that, you know, he was an entomologist who had named something. And he said, "Oh, Mark, it's really easy. All you've got to do is turn the light on at night. There's so many insects that haven't been named." Well, in Australia, that's even more so the case. In Australia, we've got so many of our insects are nowhere near being named. We know, for example, of our wasps, the head of the national collection for CSIRO is a hymenopterist, wasp specialist, and he hopes to have named 2% of Australian wasps by the time he retires. That is, we've still got 98% to name. And those of you who haven't seen it, it is worthwhile looking at. Who here has seen the fig wasp and what it does? So figs are an unusual plant in that they don't have a normal flower. They have an invaginated flower. Their flowers on the inside of a thing called a syconium. And there's a little hole called an ostiole. And that ostiole allows fig wasps to get in and they fertilize them. So the reproduction of figs is reliant on a relationship with wasps. And this is what they're like. They're sitting there in mass trying to get in through the bracts have opened, the hole, the ostioles open up, and they'll go in there, they'll push their way in, they'll lose their wings. She's already impregnated. She lays eggs. The males impregnate, the females chew the way out, fly away. And then there's a wasp that predates upon those wasps. And then there's nematodes that are on the wasps, the nematodes that are on the predatory wasp, and so it goes on. They're an incredibly complex relationship. And that gives us 1,200 species of figs that are absolutely beholden to this relationship. So figs, for example, flower all year round in terms of most of our figs, because we can't afford the wasp is dependent on that one species. So unless we've got a cycle that lasts for a whole year, it can't be an annual cycle. So things like Moreton Bay figs, you'll see the flower opening up for 9-10 months of the year. Same with Port Jackson fig. And it's why we don't have big problems with Ficus elastica seeding up everywhere, is there's no wasp for it. It's why Ficus microcarpa hilii, we don't get seedlings coming up from it in Sydney or in Brisbane, is the wasp hasn't migrated. Well, the wasp has now hit Brisbane, I've been told. But yes, it needs the wasp in order for fertilization to occur, which is, I think, pretty unique relationship.
Hey, Mark, is there just one species of wasp per species of fig?
Good question. As a general principle, yes.
Okay.
As a general principle, the best way to identify the fig is based on the wasp.
Okay.
Identify the wasp,
Okay.
and you can identify the fig. The problem is, what did I say about wasp identification? We're hoping to get to about 2% in the next 10 or 15 years. So,
yeah, I'll leave it to Mr. 2%, I think.
yeah, and look, if you look at figs, there's so many variations in terms of the way they look. This is a typical what I'd consider Moreton Bay fig with a long stem and so on. If you get down to Adelaide, this is what they call a Moreton Bay fig. And that's egg-shaped. By every stretch of the imagination, I can't imagine the same tree. The leaf looks the same, but the fruit, that's sessile. That's Ficus. I'm just trying to find you a picture of an osteo. Oh, okay. Nice. Yeah, that'll do. One before. No, won't do because it's out of focus. That's one out of the Botanic Gardens, and there's a flower on the inside again. You can see bits of the stamen, so it just never gets exposed. It's not wind pollinated. It requires that relationship with the fig, with the wasp. So no wasp, no fertilization. And, again, we're at that stage where there's just so much that we don't know that essentially we're screwed. And they're amazing trees. Figs are, if you think about it, figs are one of our more complex groups of trees. They have creepers, deciduous, evergreen, shrubs, trees, they're just complex, and they defy all the standard evolutionary logic, because you can't get that variation, of course, without the wasp, and then you need the wasp to get that variation, and so it goes on. So yeah, they are a unique group of trees. So go back to your pictures. That looks like it was probably something like a longicorn beetle. In terms of the ants, once I'd identified it, if I was happy with it, I would treat it as an exotic. If it was native, I'd think about leaving it alone. But I'm guessing to do that sort of damage, it's probably exotic. And one of the things that ants do is they're really good at farming. So, for example, in our nursery area, we don't commercially produce. We grow for unique specimens in our own interests. But when the ants start farming root aphids, it's just a pain. They do so much damage to the nursery stock. They create conditions that tend to become hydrophobic, and they farm those aphids on roots to their heart's content. And I can show you what they look like. Maybe not get it out of root aphid in one go, but we'll see how it goes. Oh, there we are. We did. So there are aphids on a root. That's the tip of a pin, so it gives you some idea of size. And the ants physically move them around. And, you end up all of a sudden with, 100 pots with, 10,000 aphids that you don't get to see until you say, well, why aren't these things growing well? What's going on? And you knock out the pot and you loosen up and pot it on, and you think, oh my, we've got a big problem. So, and they do the same thing above ground too. And we saw that in one of the shots that I showed you earlier. They're really good at farming on the surface. Here's ants farming scale. So they'll move the baby scales around and attach them where they want them. So they get the honeydew. So sap-sucking insects that feed on phloem, so phloem feeding insects, get far too much sugar for their diet. And to deal with the excess sugar, they poop it out, and the ants just go crazy on that. And so the ants give them protection in order for keeping control and keeping predators away. So again, same thing happening. This is gum tree scale, and the ants are farming them, and they're protecting them. So what you end up with is the scale population increasing rapidly, often to the detriment of the host plant. So it's it's a balancing act. You don't want to get rid of it, but you don't want to not get rid of it. You want to try and keep it to a level that is close to where it needs to be. And life gets out of kilter very quickly in plants. This is, again, ants on plant hoppers. They can protect them so well and farm them so well that you end up having the plants start to get stressed and harmed by it. And, I could go through and find probably another 30 or 40 in the collection that are like that. So does that give you there's not a magic answer on it?
No, probably not. I was more thinking, there was a few trees in that backyard that died recently, and I was just wondering if the ants could have possibly had anything to do with it.
Yeah, the answer is yes. And then you've got to weigh the up. How do I treat it? Do I treat it? Fortunately, the ant powders are highly effective at control. And if I'm guessing, you're probably not going to wipe off a rare species. But I'd want to try and do my best to identify the ant beforehand, and that is a hard process.
Where would you where would you start?
Well, the white-footed house ant took me about 6 months to do. There is a support service at a lot of the museums and DPI and things like that. So what you want is a worker. So they're a social insect. you want a worker, you want a soldier, you want to try and get a number of photographs, and then send them off to your local museum. Or so in number 1 College Street in Sydney is the Australian Museum, and they have a search and discovery section or something that you can send things into, and they'll give it a go. And all they are there is amateur entomologists. There's a really good site if you're in the northern side of Australia, Brisbane Insects. He's not an entomologist, he's an amateur entomologist, so he's not you know, an authoritative person, but he's done a really good job of collating his specimens. And so we know, for example, ants are Hymenoptera. Where are we? I can't see. I'm going to have to search it, control. Oh, ants. Here we are. Hymenoptera. So, you know, we're going to start looking at, you know, is there some physical feature that we can get that, you know, looks similar, that lets us start getting it down into a group. You know, an ant like this is pretty bloody unique. So nice and easy to figure out. You know, once you start getting to different groups, it starts to just become difficult. And then there's exotics and what we do and don't have. But there are people that you can reach out to. As I said, the place I'd start at is probably the museum, the Australian Ant Image Database. No, it's gone. Be interesting to see. Yeah, okay. So there's a text there, Australian ants. I've got a feeling we've got a copy in the library. But yeah, it's just a matter of putting the hours in. But again, most entomologists that work in museums and places like that are willing to give their information if you ask them appropriately and communicate with them at a professional level. You know, I've seen this. There's lots of holes. This is the ants. Can you steer me in the right direction? And oftentimes they'll have it done in a few quick questions and at least some indication of where to go. You might get down to genus, or you might be told that it's exotic.
So are there native ants that do damage to trees like that as well?
Yeah,
or purely, yeah.
Anything can get out of filter in the natural systems. So it's like mistletoe. Mistletoe is generally a good plant that doesn't cause significant problems, but in excess numbers, it can be problematic.
Yeah.
You know, and for that reason, fortunately, most arborists aren't treating insects most of the time. In fact, you know, elm leaf beetle hit Sydney about 6-7 years ago. There was a spate of treating them, then it slowed down. Chevaphus celti hit Australia, hit Richmond. It came in through the Richmond RAF base, as far as we can figure out. The first 2 or 3 years, celtis were just hammered. But again, they seem to have settled down, probably because there are natural controls. There are natural controls for ants, and that's another whole fascinating thing. You know, for example, some of the zombie fungi affect ants, and so you end up with ant colonies being knocked back when they get too big and too large, just through natural forces. Again, vulnerable. Fall of the Roman Empire. Okay, so to your last, was that the last one? No,
I think that was the last one.
Oh no. Yes, you asked.
Yeah. I don't know what happened with the text there, but
yeah.
That's in the same property as the ants, but that's a Banksia at the front.
Yeah, and it looks like something else is happening in the outer bark. So trees, when we talk about bark and we call it phloem, we're wrong. There's a few layers of phloem in close. There's the most functional recent one produced by the cambium, but most of the bark is produced by the bark cambium, which is not involved in the same process. It's the protective process of the tree more so than the transport process. And so that's your philoderm, your philogen, and your phylem in that order going outwards. And they're susceptible to also to things coming in occasionally and doing sorts of damage. That looks more like it's been some bigger animal, maybe a sugar glider, maybe a bird or something like that. I don't know.
Yeah.
Okay. Any other
Cool.
questions?
Thank you.
My pleasure. Any other questions from the group as a whole? No one's there, Peter. They've all gone to sleep.
Oh, wait a second, I'm here. I have a question about the Genopolis or the Armillaria. I've noticed that I've seen it a lot on the base of hakias. Is that a common thing that you find, or is it just me this year?
I don't know the answer to that, Andrew. That's I think we've looked at this, but if we haven't, let me know. Come on, give me a video. There it is. I should be able to do that on YouTube. There it is. YouTube, thank you. It'll have to give me an ad. I'm sorry about that. This is in part the work of a guy called Bo Lotto, very clever man who looked at how we actually perceive and see the world. And so you've got what you see the setup, and the question is, how well do you actually see what you see, and do you interpret things? Are they the same color, the two red arrows? And so when you ask me and we talk about anecdotal answers and they're important to us, that's how we start to come up with a hypothesis. It falls a long way short from reality, and it may be a reality, it may be your reality, it may be the absolute reality, but what we'll be certain of, there'll be other realities out there as far as the perceiver is concerned. And that's where it starts to get really complex. And that's why we want research. And that's why we want research based on good controls. And you know, my favorite one in arboriculture is Alex Shigo's wound dressings. You know, make wounds anywhere in 60-degree increments, so 6 spots around the tree. Use a random generator to pick the spot, roll a dice. That tells you what 60-degree point you're at. Then you make a hole. You put a metal plate in, you paint one half, you don't paint the other half. To decide which half you paint, you flick a coin. Why? Because all those things take away our biases that we introduce. And so Alex Shigo would say that the perfect experiment is one where there's only one variable. Now, of course, that's easy to say, but one variable when you're talking about plants, there's genetic variation. And that's why we need these replicates then. And when it comes to people, it gets even more complex because there's a thing called the placebo effect. And the placebo effect is so strong that we have to use double-blind studies. And those double-blind studies means that somebody given a sugar tablet can get exactly the same results if they know what the medication is meant to do, simply because their brain can do it. And we should really believe that when it comes to our native peoples, you know, and the pointing of the bone. You point the bone and you can kill a person. The person can actually will themselves or believe themselves to death. And that's pretty amazing. So Ginopolis at the base of the tree, usually without any form of lesion. Now, I should be able to find you a lesion in my my picture collection. S-I-O-N, but maybe not. Oh, okay, we're lucky. So, you know, something like these little inverted V's, early stages of potential infection, I worry about seeing that and a fruiting body. You know, it looks like it's occluding, but that's the sort of thing that we're, now I'm worried definitely about that. Okay, not looking good. It'd be worse if it was rising up more, but again, this sunken tissue, dead material, these are leaf lesions. We don't want leaf lesions. So that's what you're looking for. Is there something where there's necrotic tissue developed or developing? If it's developing, I'm really worried. That is, if I'm seeing dead tissue and the tissue is extending up, there's recently dead tissue. And that's where, if we're taking a sample for identification, we want that zone between the live tissue and the dead tissue as a spot that we want to test. So that's where you'll take the sample and send it off if you want to do something like a DNA identification of fungus because you don't have a fruiting body. Answer the question for you, Andrew?
Yes.
I can tell you where you'll find them all the time, and that's on the outside of the tree. That's about the best I can do.
That's what I've discovered too.
But Ginopolis does look different enough, and I get asked to describe the difference, and you know, I can't. You just see it enough. It's like, you know, if I had to describe my wife, and I say this with sincerity, I know she's got hazel eyes, but if she gets kidnapped when we're overseas, she's gone. Because I've got no idea what she looks like until I see her, and then I say, I know that's her. You know, when you see those people do those delicate photographs in a crime scene, I'm sitting there thinking, they've got to be lying, because there's no way I take that notice of anyone.
Yeah.
So when I say I see the difference, I see the difference, but I don't know really what it is that I'm seeing. I just see the difference.
Well, I did a 12-slide PowerPoint presentation on Armillaria, and I still can't tell the difference between Armillaria and Ginopolis.
Well, the good news, Andrew, is you will get there. Pea-sized pieces regularly enough, you'll find that most of what you see is Ginopolis. It's far, far, far more common. Hundreds of times more common.
Okay.
For a starter, it's got a brownie color spore, rusty brownie color spore. So they're lived. It's a good saprophyte. Armillaria is a poor phyte and a weak pathogen until it gets going.
Yep.
So you'll see hundreds of trees with Ginopolis. You'll see one tree with Armillaria. It's so rare, it's exciting when you see it.
Okay.
And if I think of Sydney, I can think of probably ones that I've seen and ones that people have told me about, probably 30 or 40 colonies in Sydney versus Ginopolis, and I can probably find you 30 trees in the western, in Blacktown City Council area that have got Ginopolis. And I reckon if I drove around for a day, I could probably find you another 30 or 40.
Yeah, okay.
So Armillaria is a rarity, and that's why it makes sense just to do that quick test. And the more often you do it, Ginopolis is a reddier color. It's not a scaly. You know, my colleagues would pick it at the drop of a hat, and they'd tell you all the reasons why. You know, I haven't seen a new colony in 6 years, 7 years, something like that. It's
if you sorry to interrupt. Would you mind repeating the quick test you just referred to?
Yeah. So the two critical things for telling Ginopolis and Armillaria apart. The first one's the pea test. Take a pea-sized piece, chew it for about a minute or so, just in the mouth. Spit it out. Nothing you can eat in the way of fungi will kill you or cause serious harm at a pea-sized piece, even not the wacky ones.
No worries.
But spit it out.
Yeah.
Okay, it's just a good practice. If it's malaria, it's chili spicy. It's hot.
Yeah, right. Okay.
Genopolis is more astringent and drying.
Right.
Young Genopolis, that is before it opens up, it can be spicy. So when I say a cap, I want a cap that's open where you can see the gills.
Yeah. Okay, gotcha.
Okay. Now the other one is the spore test, and I can show you. I can show you a spore print. So that's a spore print. Just put your mushroom down and cover it. You need to cover it because otherwise you won't see the nice lines. It'll just be a mess. It doesn't matter if it's a mess. That's Genopolis. And that's the color of it. So, yeah. and you know, that's a piece of blue napkin. So is that, would you see white on that? The answer is yes. So, you know, putting it down on that, you still see it. You still see it on black. You just don't see the really dark brown. So if it gave you a dark brown spore print like the standard agaric, you wouldn't see that on black. And yes, you get weird colored spores. So, but spore prints, again, a really easy way of doing it. And, you know, I'd encourage you, if you're driving around and you see, you know, a tree at the side of the road, grab a sample, have a look. You know, it's like any of these things, the more often you see it, the more you become aware of what it is. It's like identification. You know, when you're trying to get to identify a new species, first couple of times, you know, unless it's something really unusual. I was just in Fiji last week and working on the Barringtonia asiatica. You know, you can't miss it, you can't confuse it because it has just this weirdest sporocarp. Sorry, not sporocarp, fruit. You know, you can tell I'm doing tree work. It's serious, and I actually did do tree work over there. But yeah, that made it pretty easy to pick, you know, that it was Barringtonia. But that's not always the case. You know, eucalypts, for example, are horrible to identify. And it becomes just a question of, you know, getting used to what the tree looks like. And I did one the other day, and it was just, again, that unusual form. I can't even think where it was. That was, by the way, a great shot. If anyone could tell me where that is, I'd like to know. The good thing is it's cut down.
Grey box country.
Yeah. Well, box country, yeah, which is 2/3 of the east coast of Australia, yeah. But yeah, I walked into a site where there was an eucalyptus, and the eucalyptus was Terracornis, and it was just an incredibly weeping form. And I thought, what a delightful tree. And the only reason I could identify it as Terracornis is I'd seen a weeping one once before in the Richmond Windsor area of New South Wales, and recognised it from that straight away. Otherwise, I would have been completely thrown by what it was. So it's just about constant space repetition. The more often you do it, the more likely you are to figure out what it is when the time comes. As you know, this is one that was thrown at me uniquely by my wife, who said, "What the hell is it?" And you know, again, that was probably 4 hours on a Saturday trying to answer what the hell that was, and the answer is that's Brussels sprouts. Brassica,
really?
Yeah, preserved and used ornamentally. You know, these things don't come easily. You waste a lot of time, and that's one of the reasons why, as consulting arborists, you want to charge, you know, better than just ordinary money, because for every hour that you've spent writing a report and doing that visit, you're going to spend probably another half hour at least at some stage in your life contributing to that same answer. So, you know, if I sit there and do it at $90 an hour, I'm going to just never be happy because I'm going to be constantly earning far less than I should be earning.
May I ask a question, Mark?
You certainly may. You just like to ask another one.
Testing for Phytophthora. When I was searching online, I found a website in Victoria that had
ELISA kits?
Testing kits that looked like a rat test.
Yep.
They weren't in stock, but I just wondered, are they feasible to use them?
Yes. I mean, one of the issues with Phytophthora is in urban areas is a fairly ubiquitous fungus, and it doesn't necessarily mean it's causing problems.
Okay.
And, you know, I can think of one of the Platanus out at Richmond TAFE, for example, has an malaria infection. It's been there for probably 10, 15 years. You know, there's a battle between the two all the time. And Phytophthora is not winning. Phytophthora tends to do a lot of damage with things that have got a high parenchyma content. And the one that I can think of that fits in that category is avocado, ficus. When it gets into ficus, it really does do damage, but ficus is fairly resistant to it. When it gets into brachychiton, it just turns it to pulp because, again, huge amounts of parenchyma.
Okay.
So
there's 3 eucalypts in a row. There's been a lot of trees dying in a street close to me. They're council trees. Now there's a pine tree and 2 more big eucalypts that are on the way out.
yeah, look, and it's hard to know what's causing things at the moment. I'm still seeing a lot of damage from climatic conditions we've been going through. The excessive wets have caused problems for a lot of the ironbarks, particularly Sideroxylum, but even Fibrosa. I've seen microcorys suffering oddly.
A crabra as well.
A crabra, yeah. So the ironbarks generally didn't like the climate that's been thrown at them, and here we are in the middle of a drought again, and we don't see it, but it's bone dry. You know, as I go through the back in the tree farm, I see cracks and fissures in the ground that I could put my hand into. It's not hot, so the load on the plants is not great, but, you know, I don't know what's going to happen in the next 6 or 12 weeks, and we could find things knocking back again. Having said that, I looked at a site, and I will share these 2 with you if I can. It's the best way to do this.
Mark, while you're looking, Waverley Council, I think, put out on Facebook that they've got Banksia serratus dying right across that LGA. I haven't seen any photos, but I'm just wondering if anyone's heard anything or anyone's been out there looking at them.
I don't know. I haven't, but I do know that there are lots of plants that stress back heavily, and if we looked at our how good am I at predicting the life expectancy or the Yule or the Sul or whatever you want to play with in that group of plants, I get mocked at. I look at a tree and I think, well, that'll be dead in a year's time, and 2 years' time, you're back and it's in great condition. And the tree that looked like it was in great condition is dead. So I tend not to want to try and guesstimate those things. I was on Dangar Island, Derek, about a year ago giving a talk, and the community took me around, and there was this massive decline going through the casuarinas, and they'd need to contact me recently. And I said, how's the casuarina decline going? And they said, halted and just everything went back. The trees flushed up and, you know, continued on. And I thought for sure we had a new disease that was going to hit quite badly, but it stopped and slowed.
Yeah, you're quite right. That transition between wet and dry, or dry to wet, and then back to dry again, seems to be knocking a lot of trees around.
Yeah, but you don't know what they're going to do longer term, and that's the
No.
unknown quantities. Sorry, as I try and find my way around.
Is this a free maps program?
No, this is not a free one.
No.
It's a ridiculously expensive one, unfortunately.
That's why it was such good quality.
Yeah. This is near maps, and they take photographs fairly regularly. I'm just trying to find a school.
If you are looking for something similar to near maps, I've recently, so I used near maps previously, but like you said, like they're crazy expensive. I've actually been using Metro Maps for the last 6 months, and it's still a paid service, but it's probably about half the price of near maps, and it's not as functional, but the imagery is I'd say pretty much as good. Quite a reduced price if you are looking.
Yeah, Metro Maps.
Yeah, it's got a
Metro
near
Maps.
infrared imagery that's used, and that is really good. Near infrared is good for picking plant stress, and so it's a really useful tool. And you can also get yourself a standard digital camera set up for near infrared photography and do the same thing. And NASA makes a nice pair of glasses that I've got a set of that just make your head spin otherwise. But again, you can pick up early stress using the glasses. This is a school that I looked at, and I got asked the question, what's our risk like? And here's the school being used. You can see some kids. If we look at it, there's some out here, there's some going through here, but it doesn't look like that's a lunchtime. And there's a bit of a long shadow, so I'm going to say it's probably not. So we want to try and get to a stage where you can see how few kids are in this playground at any one moment in time. Can you see that? Kid. This is a school with 500 students. And I spent a fair bit of time going through looking at aerial images, trying to find out periods when the students were outside playing, because I wanted to get an idea of how the area is used. And what I found, most amazingly, is that the playground area of the school was used had an occupation rate of less than 1,000th. That is, the students are out there for 1 hour in a day, but they occupy less than 1% of the space to start off with. And I don't know that we often do that.
Sorry, I was just going to ask what you're using for your occupancy calculation there.
I just looking at the area, so I measured. I sat there and painstakingly measured all the external areas, and then looked at the number of students that were in that area, and then said, how are we going in terms of occupation? And every time you see them out in the, okay, there's a heaviest use I think I found. And you say, well, look, there's not, we zoom in 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. There's 20 just in that area there. There's probably another 30 here. There's another half dozen here. We've got a COLA, covered outdoor learning area, another one here, typical Department of Education, knock down trees, put up COLAs. We've got another few students here. Obviously, we lose things under the trees and the shadows, but realistically, how likely do you think that is going to have 100% occupancy in any of those areas? If I just took that area there and measured it, and then counted the size of the student, which is less than 1/4 of a square meter.
Then you've also got the fact that kids aren't at school at night time and all that other stuff.
That's right. So you look at the fact that you've got 240 hours of occupation in a year, and then less than 1% occupation of that area. And it worked out that, you know, with a slight allowance for weather, because they're not going to be out during inclement weather, there's less than a 1 in 10,000 occupation rate for in the high use areas of the school. And what was fascinating is this was a risk assessment that was done by a third party, who then said these trees here had issues that you need to deal with, and these are getting down to the 1 in 100,000 to a 1 in a million occupation rate.
Too broadly acceptable.
It's acceptable even if the tree was almost an imminent likelihood of failure. Yeah, it was an interesting exercise to actually sit there and see how's the play area. I sort of envisaged this really thick concentration. Now, this is a big school and low student numbers. 500 for a school of this size is not that great. I get some of the inner city areas where, you know, they've got 200 students and that's their play area, and that's obviously going to be a lot higher. But it's to sit down and do that task. I hadn't done it, and it really did make me think about what I was doing. And
really interesting. So do you normally take up that method when undertaking sort of risk assessment for a school, or do you tend to stick with the track QTRA type?
QTRA, well, that's a QTRA function. So I was trying to get, rather than my perceptual data, the grey, I wanted to try and find out if there was some way I could support the fact that, you know, this was less than a 1% occupation rate, or less than 0.1% occupation rate, or 0.01, which is what it was.
Yeah.
Could I argue that based on observable data? And so this is the heaviest use I find in the school in the production of the near maps. And so I'm pretty comfortable that the occupation rate is not that high. And that's because there are kids that are under the collar still. That's because there are kids that are, not using the area the way that we think they would. And there certainly will be, no question about it, students hidden under the shade of the trees. And I know that because we get wear marks underneath the trees, and that gives you, again, an idea of how an area is being used. So if you go to a school and you see the grass missing under a tree and it's all compacted, you can be fairly certain that students are using it. But the real bit that I found obscure was, I would have estimated this area as probably 1 in 10,000, not this area.
Yeah. So when you're stepping outside of those sort of predetermined sort of ranges that you get within QTRA, etc., do you feel sort of comfortable with your report writing? Well, obviously you do, but I mean, how is that just a case of showing your workings out? In layman's terms.
And again, I don't necessarily have to show it. I certainly calculated. I screenshotted it. Once I calculated the area, I screenshotted this image. I went in and zoomed in on some and shot areas, and I was pretty comfortable that, if I looked at this area here, I can see some students there. I can see some students here. I see a bit of a tree cover here. That's Amelia there. I know that. And this is August, so that makes sense. But, if I were to take all those little bods there and cramp them all in, they're probably going to fill an area this big. If I got them all, if I said to the teacher, line them all up, sit them all down, shoulder to shoulder in an area, how big an area would that take up? And the answer is not a very big area. And that's, as I look at this, under risk assessment, we're not asked to do everything and anything to calculate risk. We're just asked to do what's reasonable. And so if somebody had said, look, the occupation rate here is 1 in 1,000, I probably wouldn't have argued it. If they said it was 1 in 100, I might have said, well, I think you're probably being a little bit conservative, but I'm probably not going to argue it.
It's just
I would now, because I've sat down and done the exercise. And now that I've done the exercise and having shared it with you, it's an exercise that you can do, and it doesn't have to be as sophisticated as this. Google Earth Pro is free software.
Yeah, no, it's really, and apologies, I've hijacked the conversation
No,
a
look,
little,
it's a
probably
reasonable set of
more appropriate for yeah, probably more appropriate for the writing Alvarez
no,
reports. But
no,
yeah, no,
look,
I just find it interesting because with relatively incredibly limited experience compared to yourself, I tend to be very regimented against the sort of methodologies that are spelled out by QTRA or valid or track or whatever. So, you know, always a bit scared to sort of step outside of those predetermined sort of ranges. So yeah, apologies, but it was really,
no,
really
look,
interesting to see.
QTRA does have a way of dealing with it by assuming random usage.
Mhm.
And then looking at the size part. And I wouldn't normally sit down and do that, again, because I'm super slack and lazy. But just to be able to, I can't put the same one.
And Mark, your results would be very dependent on the probability of failure of the tree, though, even though the occupancy rate could be high.
Yes, that's right. The three things that determine the end output of risk, and the fault with the system that was used in the school originally was that the system that they used had likelihood of failure and consequence, and that's the standard risk matrix. Okay, the problem with it is we're dealing with mobile targets, so it's a likelihood of failure times the consequence times the likelihood of the target being there. And that's why track has two matrices, because you can't have a three-dimensional matrix that works in the human brain. That's why QTRA uses a three-wheel calculator. That's why Valid has three inputs. And so they'd done this two-factor input and had tree work coming out of their head. The school was mortified about how much work they had been having to do. And I got
It's
there
a
and looked
fairly
at it.
well-known company with their own matrix.
Look, there's a whole association. The Tree Contractors Association has a document that adopts that exact same policy. And yes, it's commonplace because people don't understand risk
Yeah.
at the right level when it comes to trees. And there must be the three factors. The three factors have to include. See, if I'm driving a car and I'm speeding and I'm drunk, I can calculate the likelihood of me being hurt in that car because I can't get out of that car. But here, a person's not necessarily under the tree. And so that's why we had this three component. So yes, they had overestimated the risk substantially. And I just wanted to understand from my side, you know, what the real usage was like. How far had they overestimated it? And the answer is, in the main use areas, they'd overestimated it by a factor of 10,000. And in the low use areas, by a factor of 1,000,000. That's
Sorry. Do schools have any
overestimate.
data on their occupancy? Not really.
Yeah, well, we asked them the number of students, the number of
Yeah.
staff.
Okay.
So we collect that information, and that gives us an idea. But again, depending on when I'm there, if I'm there, I'm doing 5 schools tomorrow. And at least 2 of those I'll be there during the playtime. And you very quickly get an idea of the density again. And I said, my assumption has always been that it was probably a lot higher than it is. I think if I went down and crunched it for more detail, I would probably find that I'm still overestimating the occupation rate. I guess that was the advantage of sharing what I've just shared with you. And it related because, so this is Google Earth Pro, and Google Earth Pro is free. And down the bottom here is the historic bar, and that historic bar opens up the top here. And every time they've done a shot, you can go back and see what that's like. And so again, you get multiple images that you can go through and have a look at. And that's a freebie if you want a freebie one. And what I am pretty certain of is, for example, a tree like this here, what's the real risk? What's the target? There is a target. I actually know the specific target. this tree here, again, it's in the parking area. So the only people that come in here and are exposed to harm by this tree is the GA. And he's using a zero-turn mower, and he's traveling at better than 5 kilometers an hour. Now I can really start calculating it out. Okay. So his exposure to mow underneath the canopy of that tree is probably, you know, less than 2 minutes a fortnight, because he's not mowing every week. And so you can start to realize that the occupation in these areas is really small. Does that help? And hopefully you can see how you could just go back through it and see how an area is being used. I raised it because the other thing that went with it is, you know, if I can type it in, Blackett Primary.
Is that report something that you might consider discussing in one of the Writing Arborist reports sessions, or is it too commercial to?
Only to have it.
That'd be awesome.
We've discussed risk a fair bit at these, because it's a, unfortunately it's an over exaggerated area that we get caught up in. And it's not that that's bad, it's just that that's the reality, and as a result we tend to get anxious about it. What we should be doing is sleeping very well at night, realizing that the assessments that we're making are based on such rare events. 5 deaths a year in Australia. You know, I should feel criminal charging a school to do a risk assessment. This one won't work this way because you've got to go back and look at the historic shots. But I want to look at this one here, so I'm going to go back and I'll do it on Google. So that was Blackett Public School, Blackett, and it was one that I did today. And again, I thought, how are these trees going? What's affecting their health? How are we going with it? So Street View is a wonderful piece of software that's offered by Google, again for free. And I can go and look at trees historically. So that tree is not looking too good. Will we agree with that?
Yeah.
Eucalyptus microcories. And that's May 2021. But I can see more dates. And I've just dropped you guys down the bottom and I want you up the top. So what was it like in 2020? It was pretty crappy in 2020. How about 2019? Pristine. Yeah. Okay, so the question is, what does it look like now? And I should be able to answer that. I'm going in here and typing in Blackett, Blackett, because I went and looked at the tree and did an assessment, and I was trying to figure out how badly stressed the tree was. And that's the tree there. It's got one big dead branch. Overall, it's not doing too bad a job of starting to recover.
It's like it's gone through cycles.
Yeah, if I had to predict, I would have said the tree's going to die on that 2019 image. 2020 image. I would have said, no, it's gone. I look at it now and I think my thing was, if it dies, remove it. If it recovers, remove any dead branches greater than 10 centimeters in diameter from the tree. That's it. A low use area, it's in a garden bed, not generally used by the students. It's not a risk issue, it's a tree maintenance issue. Why would you do that? You know, tree's valuable. Take care of it. Less paperwork, a lot less paperwork saying there's no branches greater than 10 centimeters in diameter that are dead in the school than there is filling out the paperwork and saying I've assessed it somehow or other, compared it with the last time I visited, which is what the DEC has in its checklist form, and the principals are meant to do, and I can't do it. I look at a tree and I say, has it got worse or better since last year? I'd have to sit down and look at photographs and compare it and then adjust for light, adjust from the angle that I've taken from. I can't do it. I give up. So I look at it each time with a new set of eyes and say, am I worried about that tree at this stage? No. There was a tree there that I was concerned about. I guess you could have said the risk was probably unacceptably high from that. I didn't think it was something that I need to worry about. No, not there, to go the other way. I went past it. That tree there, I pushed it over. I load tested it, realised that it was nowhere near, you know, 3 kids could have pushed it over maybe. I was able to, so, you know, this is Tongan Samoan territory, you know, 3 refrigerators in the thing. They're bigger than me probably, might have pushed it over. But generally, you know, if I can get it to the stage where I think it's going to go, I'll push it over. What's the risk associated with that tree now?
0.
No, there's a trip hazard there.
Oh, yeah, until it's cleaned up.
Yeah, I was about to say, did you clean up the trip hazard? Yeah.
No, Derek, I didn't. But that gives me a lot of confidence as I push on trees. You know, if I can't sort of make them rock a reasonable amount, wind's not going to make them rock. So now I'm not looking at whole trees falling over. I'm looking at these little bits of deadwood falling. And, you know, when are they going to fall? Mostly during inclement weather when no one's going to be around again. If there was a seed under there, I'd feel different, you know. But what was the likelihood that a student was going to be present when the tree got to that level? The answer is I had that tree at 45 degrees before it started to go by itself. So if little Johnny's under it and it's at 45 degrees and no one's picked it up, little Johnny needs to die because he's stupid. No one needs genome breeding. Not literally, but you get my point. You know, there's a point to which we've just got to say stupid is stupid. And, you know, one of our tree fatalities in the school relates to exactly that. I won't mention the school out of respect, but a tree had a branch drop out. It was leaning against the trunk of the tree. Little Johnny was pulling on it, pulled it backwards. The stub end went backwards and hit a kid behind him and killed him. You know, that's not a tree death. That's stupid death. That's a bad supervision death. And little Johnny should never have been able to play with a branch. And I feel sorry for little Johnny because, you know, he'll live with guilt over that. So we probably should take the teacher that was on duty and get another tree and fell it on them. Not literally, to get the point. So again, it was just good to see that what you think's going to fail and die doesn't. You know, we're not good at predicting those things, and particularly given that Australia is a country of extremes rather than the English predictability of, you know, oh, we've gone a week without rain, we're in the middle of a drought. You know, we don't know where we are. We lose fact of the changes in climate. That how dry, for example, it is in Sydney at the moment is something that Sydney-siders won't be largely aware of because we had such a wet season 18 months ago. Well, 18 months ago is not now.
Yeah.
And I'm guessing that all of you find that same thing. It's like when you ask a client and they talk to you about the tree and they say, oh, no, that tree, oh, that was that big 20 or 30 years ago. And you look at it and you think, no, I know it was a lot smaller. Perception starts to take over. So if there's one thing you can take away from today is that when you're trying to interpret things, remember that you're perceiving it and you're a perceptual individual. And so there'll be error in the way that you see that. And I'm comfortable with saying that when I'm doing risk assessments, I'm missing things, I'm not seeing things perfectly. All of those are just so irrelevant in general to the end process, because the things that we really need to be looking for are things that an 8-year-old child will pick up. We won't be missing. You know, this pedantic, you know, is that a really tight union there, or could that fail? We really got to stop fixating on every possible item, except where you've got huge occupation rates, and that's in very small parts of the city. And those of you who haven't done valid training, David Evans does a really good job of trying to give you some good benchmarks in terms of road usage and pedestrian usage that lets you see what it looks like. So if you get a chance to do that, it's worthwhile doing. Any other questions before we call it quits for the day? Right, guys.
Not relevant anymore, Mark, but I sent you a spore print of the Genoplis from my tree from way back when. I promised you a spore print. I've just sent it while we're talking about it. But
and it was nice and brown.
Oh, it was rusty as. It was the best news I've ever got.
Yeah,
I did a little dance for joy.
And Mark, that's why I say that it's important to just keep on doing these things until you're getting to the stage where you look at it and you laugh and you say, "Oh no, it's Genoplis straight away."
Yeah.
And that's just constant space repetition. It'll eventually get to the stage where you look at it and you say, "Ah, I know that." And I remember the last time I saw Armillaria, I walked down the driveway to look at a different tree altogether, and I said, "On the way back, we're going to talk about your Armillaria infection." And it was that obvious to me that it wasn't Genoplis and that it was Armillaria. And so that was the first time I really got to the stage where I thought, yep, I've got it down so pat now, I can't get it wrong.
Yeah.
Now, I probably will because, you know, like I said, there's that lovely Pterocornis the other day that was nice weeping form. And if someone reminds me next week, I'll pull it up and have it ready for you. And, you know, there's huge variation in plant material, there's huge variation in fungal material, and it's so much so that, as you know now, identification is generally done using DNA. The one caution I'll give you in terms of the use of test kits and DNA for identification is just because it's present doesn't mean it's guilty. If you did a DNA test, a mass PCR test down at Homebush, because there's been a murder there last week, you'll probably find some of my genome there from 3 weeks ago. It does not mean that I killed the person, okay? And so you've got to look at more than just the presence of the organism, and that is the organism actually doing the damage, or is it just there as a secondary or an opportunistic situation? And that's, again, a matter of looking at symptomatics probably more than anything else. Guys, I'll speak to you next week.